예, 오후 세션인 두 번째 파트 넥스트 드라이버스
Ladies and gentlemen, we are now going to begin the second session this afternoon under the topic of next drivers in ADC and immuno-oncology. And the first project will be delivered on highlights of LCB's emerging ADC pipelines. And this first presentation will be given by Dr. Cholong Jung, Head of R&D at LCB. Good afternoon, everyone. I'm Jung Chorung. Unlike this morning, all the presentations will be delivered in Korean, so hopefully communication will be better. So many technologies have been explained this morning, so I'd like to talk about a brief introduction of technology, as well as target selection and payload selection, and what kind of ADC products we are having in the pipeline. So I think that you have a good understanding on the concept of ADC. So ADC is antibody and drug conjugates. And our technology contains linker technologies and conjugate technologies, which is the foundation for our technologies. And then our technologies and product have advanced, and now our focus has been expanded to include antibodies. So you have heard of the excellence of linker system. So the first generation of ADC technologies, as shown in Seattle Genetics and Immunogen. So there is a mixture of one antibody and different types of DR. So we want to improve heterogeneous property. And sometimes the efficacy is low, and sometimes there is a problem with toxicity. So we developed this product, and we went through clinical trials. So we wanted to improve the efficacy and toxicity with our linker payload product. And ADC need to deliver the drugs, and that should be internalized in the cancer cells. So there is a pre-payload in the cancer cells, and then DER is being lowered significantly. So our purpose is to deliver payload to the cancer cells, and this function of ADC is not working properly, and then the antibody has loosened toxicity. That's why it did not have the intended effect. So we need to improve conjugation problem so that we are able to toxicity and tolerability issue. That is the background behind our product development. And if we look at approved tox profiles, it proves that our assumptions are correct. Here you can find 4 ADCs which have already been approved. And all of these have different targets. They are the targeted therapy antibodies. But as you can see here, all of them have same numbers. That is not based on target. All of them are using same MMAA and same linkers. That's why toxicity profiles are same. if the linker is the cause, then if we make changes to the linker, toxicity profile should be different. So that we apply technology making changes to toxicity. And toxicity profile has improved regardless of target. And the monkey toxicity was not target dependent. That was based on platform. That was based on the linker payload platform. So when we analyze other ADC data, for example, there is no improvement in the MED. That is because they are driven by platform. So our linker technologies developed by our company can implement original ADC. Why we are having this kind of toxicity profiles with linker technologies? This data was presented already in the morning. So our ADC is stable, but look at the competitors' products, then 3 out of 4 are put into cells, cancer cells, because these materials are causing toxicities. But ADC has only 1 out of 4. So antibody cannot be effective into the cancer cells. So these drugs onto the antibodies are not good enough. And if that is same antibody and same payload and same linker, if ADC, our link is connected, it is much more effective. And our competitor is using 4 MPK, but ours is 1 MPK, but competitors is less effective than ours. So it is the linker payload makes a real difference. But as you saw in the diagram, 1 out of 4 is lost in the traditional ADC. That is the difference. So through this kind of preclinical trials result, we secure differentiated data, and then we are confident that we can bring benefits through patients based on our platform. That's why we are planning to expand our pipeline. We are not just relying on the linker technologies in order to expand our pipeline. As mentioned by our CEO, unlike other companies, so we are GMP ready and clinical trials ready with a variety of payloads. We are not randomly following. We are thinking about optimal combinations depending on target indications with less toxicity. So we are evaluating on-target and off-target toxicities in order to identify optimal combinations. So also the immune modulators will be developed based on the antibodies. These days, many companies, including ours, are developing bispecific antibodies in order to be cancer selective and in order to have more efficient drug deliveries. So we are working on antibody engineering. We are trying our best, like by investing lots of time in order to achieve optimal combinations. So including this kind of pipelines, preclinical trials, as well as discovery projects are also being worked on. When we select various programs, what is very important in selecting targets? There are many different considerations because we work on a variety of projects. So there are the existing projects and new projects. We'd like to make a differentiation. So as you can see here on this cancer, in the case of lung cancer or breast cancer, so there are known targets and there would be known new targets. We want to see whether they are dependent or independent or exclusive with each other. Different expression is not always good. So we want to identify patient groups with medical unmet needs. So we look at target expressions and the mutations. So we can refer to known databases. So there would be validated targets through known databases. And also we can utilize spatial transcriptomics to see whether genes are exclusive in patients or whether they exist in different cells or in the neighboring cells. Through that kind of analysis, so we can make decision how to develop ADC, whether it would be better to be bispecific antibodies or not. And also we consider what kind of combinations would be most efficient for the patients with minimal toxicity. So we do the analysis at a protein level as well as at a combination level. So based on the organ level indications, we are also preparing for backup pipelines. So due to time constraint, I cannot show you all of the targets today. So this morning, as mentioned by Mr. CEO, by the end of 2027, about 20 IND applications will be submitted, which is our goal. So apart from the existing clinical trials projects, many projects are being planned for IND on an annual basis. For example, Cloud 18 was presented, and today SICM5 will be introduced, and also STING antagonist will be introduced by its Chang. As for the SICM5, so we are targeting best in class for the solid cancer, solid tumors. That is exclusive to Part2. So that we are targeting patients with unmet medical needs. So our target is lung cancer and the colorectal cancer. So there will be more elaborate explanation on targets by the next presenter. Normal expression is pretty limited. So that is very good ADC target. and also reverse correlation with Part2. That is observed very well in NSCLC, meaning that there would be high demand in the medical field. So
we have introduced differentiated antibodies based on the research by Professor Jo Byung-chul. Just like what we have shown in other links and payload technologies, and we are trying to achieve best in class. Then what kind of payloads are the best? TOPA inhibitor, micro inhibitors were used, and we are testing many different efficacies in many different models, and we have actually selected them. This article is well known, and the competitor drug is like U6MP. In our trial, it's a topo drug, and it's amount is 3 millimeter. So at the same dosage, the efficacy is better for our drug. So at a lower amount, we show this much efficacy. That means it has really great synergistic effects. So we can actually combine these drugs to produce a best-in-class ADC. So regarding targets, later the speakers will give you more details. Now let me talk about next drivers. The bispecific ADCs. We actually have CD22 and CD20. This is a BCL and BSNHL, and it is targeting them. So we are targeting these patients. CD22 and CD20 are very well-known targets. CD22, for CD22, you already have Besponsa in the market. And for CD20, the well-known drug is the rituximab. It's already approved, and it is used as a basic regimen for R-CHOP. So we are trying to expand our ADCs in this type of diseases. As you see here, CAR-T is gaining a lot of popularity and a lot of trials going on that. CD19 CAR-T achieved a lot of clinical success. So CD19 reduced and CD22 is increased. So according to many articles, we see that kind of outcome. So existing drugs couldn't be really effective for certain patients. So for these patients, we decided to develop bispecific antibody for CD20 and CD22. So we are the first company who is targeting these two. So we are going to be the first to enter the clinical trial with that, and also we will be the best in class. We have great technologies, we have great platforms, so we could just choose one of them. But why we are trying to achieve more difficult one by specific ADCs? So their expressions are not consistent depending on patients. For certain patients, CD20 and CD22 are disproportionately expressed. sometimes certain cells are more expressed than the other. So you could develop the monoclone antibody ADC, but there must be a patient with residual disease. That's why we are targeting bispecific antibodies. Its targeted indication is non-Hodgkin lymphoma and B-cell lymphoma. And DIB cell is also one of the targets. As I said earlier, the patients are showing heterogeneous expressions. Look at this graph. This graph is about only one patient. So you see the B-cell lymphoma, and we analyzed with PACs with CD1, CD20, and CD22. So on the left side, you only see the CD22. In the middle, you see both CD22 and CD20 are expressed. And on the right side, you see only CD22 are expressed. So basically, patients show this type of characteristics. That's why we came up with an idea of targeting the two. So only CD22 has developed ADC. So there's no ADC for CD20. So we believe our answer is bispecific ADCs. So we believe the bispecific ADC will be more effective than CD20 ADC or CD22 ADC. When it comes to B-cell lymphoma, we saw how effective it will be compared to the combination therapy. At least we thought it will be the same or equivalent. So when we administered a single time, and it was really effective. When we did a PACS analysis, there are many different combinations of expressions. So most patients have these characteristics. For these patients, bispecific antibody has great potential for ADC. Some patients show only CD20 expression or CD22 expression alone, but for patients with both CD20 and CD22 expressions together, bispecific antibody could be a great solution. So as I said earlier, so we are currently developing twin ADC, but right now there's a CD22 ADC approved, but given all the indices, I think our drug will be the best in class. So our platform is the only one that shows really great PBD-based safety profile. So I think in terms of TI, we will be the best in class. So we are developing not only linkers but also payloads. As was mentioned by the previous two speakers, recently, topo inhibitor, there's a great aspirations for a topo inhibitor and pipeline explosions. There's an increasing number of pipelines for topoisomerase inhibitor. So many ADCs with topo inhibitor will be approved or have already been approved. So, which means that patients will be more exposed to TOPA inhibitor-based ADCs, and it will actually increase the resistance of patients to TOPA inhibitors. So there's a great potential for non-TOPA inhibitor ADC. So we need to prepare a new payload with new mode of action. So we are conducting research on that. So in order to overcome resistance, what type of mode of action is required? What kind of drug it will be? We cannot come to conclusion quick because it will be differ by patient. So we are researching new payloads a lot. So we are trying to develop new topoisomerase inhibitor-based drug. So ADC's resistance is involved in the resistance to the drug itself, but also it involves the less delivery of drug. So 4 or 8 should be delivered to the tumor, but only 1 or 2 gets delivered. So by changing the linker system, such resistance could be overcome, or more potent or more potent or new scaffolds can be utilized to overcome resistance. So among the microtubule inhibition mode of action drugs, we are trying to develop best-in-class. So modulating agent-based payloads and new mode of action payloads. We are trying to develop these. We could do this by our efforts alone, but sometimes we find it impossible. So we are trying to utilize many different resources, including partners outside. So through many collaborative projects, we are trying to develop new payloads. So that way we will achieve innovations. So we started with the linker, but in the payloads, we are also pushing forward with innovations. I wish we could show the new topo I related data, but right now we are at the preclinical stage. So we don't want to run the risk of exposing TPP. So hopefully next year we'll be able to show you more detailed data. But briefly, I could say this much. This is not the existing scaffold you see in the market. And we will develop more potent drug which can overcome resistance caused by the existing compounds. And one more thing is that I said we started with linker, but we made a great leap forward, and we are not complacent with the existing linker system we have developed. Therefore, so one of the questions regarding our platform technologies patent, the patent might expire in 10 years or so, then what is our next step? So we are currently working on the next step. We are developing new linker systems. And also we are going to adopt the low DAR strategy. High DAR can actually worsen PK profile because it can increase a non-specific update. So we will transition to low DAR to reduce non-specific uptake, and by adjusting many different linkers, we will try to control hydrophobic properties of drug. So in order to reduce the one specific uptake, we are trying to develop a more stable, more safe, and more effective linker system. In the near future, we'll be able to disclose our data about that. So it's not this year, but maybe in the future. So linkers technologies and new payload technologies that we are developing will continue to improve our patent profile portfolios. That way, we are going to expand our business values. We started with conjugate technology, but we are also working very hard and innovating in the targets and payloads. With this, I'd like to conclude my presentation. Thank you very much. The Q&A session will happen after Professor Chang Suk-young's presentation. Now, I'd like to invite Dr. Jo Byung-chul. He's going to make a presentation titled Revisiting the Dogma of CCAM's Directed ADC. Professor Cho is a professor in oncology at Yonsei University of School of Medicine, and also he is a center of the Lung Cancer Center. He is a great authority in the oncology field, and he has led more than 60 clinical trials, and he's played a critical role in lepotranidine laser amivantamine drugs. Thank you very much. Thank you very much for inviting me to this great occasion.
So in my talk, I'm going to talk about CCAM5. So why the LigaChemBio need to develop ADC targeting CCAM5? That is the topic of my presentation about myself over the past 10 years. I've been working on the ADCs and clinical development and preclinical trials for both local and global pharmaceuticals, and I have learned some lessons. So when I go to conference on the ADC, many people say ADC is not Lego blocks. It's like the antibody linker payload 1 + 1 + 1 the form of a Lego block. But if we make it well, it could be 1 + 1 + 1 would be equal to 6 instead of 3. So that is very promising. And I also felt that during clinical trials as well as preclinical trials, and we've done research with various types of ADCs in my research center. And LCB's the platform is very outstanding. That is one of the lessons I've learned with my prior experiences. And the lung cancer TROP2 ADC has been developed a lot. Some companies have achieved a very modest success. However, they were withdrawn from the FDA approval. And for a Chinese company with the same TROP2 ADC, and they got very significant clinical outcomes. Even for the same target, TROP2, even for the same ADC, depending on what kind of platform they are using, they may have different clinical outcomes. So today, I'd like to talk about CCAM5. So I'd like to talk about different approach by LigaChem on this. I'd like to briefly talk about Ta'an. Some of you may know already. We have done many translational research for the past 20 years, serving as a hub for the cancer-related clinical research in Korea. So we have about 20 researchers, and we also working together with about 54 PhD degree researchers, so that we have lots of experiences on different types of drugs. So I have identified those unmet medical needs while I worked on the cancer-related translational clinical research. So we joined hands with LigaChem to develop CEACAM5 targeting antibodies. Antibody, linker, and payload should be combined in a very optimal way so that there can be minimal toxicity with the patient while having maximum efficacies. So there was a great boom initially about ADC. So Ho3D, XD, and Dot2D XD. Many drugs were developed, out of which some of the drugs became blockbusters, such as NR2 became blockbuster, but other drugs ended up with as a failure. And the common aim would be increasing efficacy while reducing systemic toxicity to enhance therapeutic index. There are a variety of criteria. A tumor to normal ratio should be good, and also target antigen receptor is also important. So there are certain characteristics to be successful. But it is very hard to find the optimal combination of linker and payload. Why I think that CEACAM5 is a good target. So we are going to refer to public database, of course. But also, as you can see here, we have collaborators. Internally, we have accumulated patient database and annotated clinical samples and single cells and bulk cells for target discovery. So we are using this kind of database to prove that CEACAM5 is great target. We targeted 100 lung cancer patients. This is a single cell RNA sequencing. Please take a look at red dots. That is specific to epithelial cells, especially TKI refractory patients have a high level of expressions. And also, as you can see here, CEACAM5 high patients have lower survival ratio. And also at the protein level, we have about 100 lung cancer patients, and as you can see here, tumor to normal ratio is pretty good. And also at the protein level, TKI refractory patients are pretty upgraded. And not just lung cancer patients, but also colorectal cancer patients have a good normal to tumor ratio. So we created radar plots to prove that these are the great targets, including internalization and efficiencies and other parameters. And we got to know that the CEACAM5 is great ADC target. So this is Sanofi's data. As you can see here, colon adenocarcinoma, stomach adenocarcinoma, and CEACAM5 has a great tumor to normal ratio and specific to tumor epithelial cell with a high expression. There are the various biologic evidences. CEACAM5 can be expressed in epithelial cells, cell polarization, and the cell architecture are also affected. And not just tumor intrinsic, but also tumor extrinsic mechanisms, especially immunism mechanisms, can be modulated. So when we engage that as ADC target, then we can increase therapeutic effects based on a variety of biological data. Why do I really think that CEACAM5 is a great target? So as you can see here, tosametabe-ravintenzin, CEACAM5 ADC of Sanofi. I was the lead author of clinical trials from phase 1 through phase 3. So I was engaged with the CEACAM5 ADC development directly. That's why I know what's good with this and what kind of additional potential we may have. So I'd like to briefly talk about my own private experiences. So tosametabe-ravintenzin is the Sanofi's developed one that features humanized antibody specific for CEACAM5, and also as for the cytotoxic agent, it used TM4. Looking at phase 1 and phase 2 data, overall population is like a high expressors and moderate expressors. And ORR is about 20% in high expressors. That is pretty promising, and not just response rate, but also with a reduced tumor size. Half of the patients have not reached PR, but they have shown tumor shrinkage. Another factor I'd like to mention here is that if you take a look at the swimmer plots, many patients are not showing partial response, but they are remaining with a stable status. So these are the clinical benefits. The key message I'd like to talk about, half of the partial response patients have maintained PR for more than 1 year, meaning that this medication has very durable benefit. Based on promising data of phase 2, phase 3 was conducted. And out of CEACAM5 positive data, so the standard of care was approved in the 1960s. Compared to the standard of the care, clinical trials were conducted. And dual primary endpoints were PFS per IRC and OS. Unfortunately, they did not show significant achievements in terms of dual primary endpoints. But one thing encouraging here is that I told you about durability of this drug. Blue curve represents long-term benefits, which is pretty encouraging. About 20% of the patients shown PR, partial response, and the durability of the patients of PR was 11 months, which is pretty good. So this drug has some limitations, but if we overcome those limitations, it can be successful clinically based on this table. And another limitation we need to overcome is, if you take a look at this data, CEACAM5 tumor expression, if expression is higher, there could be higher benefits in patients. And soluble CEACAM5, if soluble CEACAM5 is high, it may lead to poor performances. So we need to develop antibody which is not bound to soluble CEACAM5. And one thing very interesting out of this data is in terms of adverse events, such as dose reduction and discontinuation, that was pretty low. However, those delays because of adverse events were higher relative to docetaxel. I'm gonna tell you more later on. That is because of DA4 related toxicity was not recognized at the time of development. That is very horrible ocular toxicity that can be generated to patients. So people may become blind because of this toxicity adverse event. So these are the issues we need to overcome, these limitations. About 30% of the overall patients may have these adverse events. And the patients and the doctors would be scared, which may lead to low the complacency. So I was engaged with CEACAM5 directed ADC clinical trials. So I think that as long as we overcome these limitations, it can lead to success. For example, because we are talking about refractory patients, so we'd like to improve response rate more. So CEACAM5 is antibody, but we'd like to make it with better internalization. Even for moderate expressors, there is certain level of activities. So affinity should be good. Avidity should also be good so that we can deal with not just the high expressors but also moderate expressors. Third one is CEACAM5 plasma shedding. So that may have a negative impact on the patients. So it should not be bound to shedding plasma. The fourth limitation is about ocular toxicity. So usually doctors cannot bear ocular toxicity. So as long as we overcome these limitations, things are pretty promising. So our goal was to develop antibodies with these characteristics. We developed many clones, but recombinant CEACAM5 or high affinity, or the CEACAM5 clones which are not bound to soluble CEACAM5, or with a higher level of internalization, so they can have higher or better activities for high and moderate expressors. So again, that is very important not to be bound by soluble CEACAM5. So we used various patient samples to reduce binding. That was proven by different types of data. So less binding and better internalization. So we compared our antibody against the tuzumab. For the therapeutically clinical higher doses, binding is better, or affinity is better. So that compared to tuzumab, our antibody has better binding abilities. why we are expected to have better binding or better affinity? We compared antibody characteristics. Tuzamitamab is bound to A3 and B3 at the lower end, but our antibody has binding, the multiple binding abilities to both the higher end and lower end, which lead to enhanced binding capabilities. This is another data. The antibody has dual binding characteristics, so it can have better affinity, and also even through epitope mapping, it shows better binding. Why it has the dual binding, including A3 and B3? If we take a look at sequencing, so the antibodies bound here can also be bounded to the lower part. That's why it can have better affinity. So this kind of characteristics of the antibody can be associated with higher internalization in theory. And as you can see here, one antibody can be bound to antom and A3B3, and when the dosage is higher, then that can also be ended to other lower ends of CEACAM5, which can result in better internalization. We conducted the various in vitro and in vivo experiments. If you take a look at this table, we used LZB platform. Compared to tuzamitamab ravtansine, our antibody has a great tumor to normal ratio. It is lower in normal cells but great in tumor cells, and in vivo benefit with the same dosage of tuzamitamab ravtansine. The ADC has even greater effects in vivo data. Global development is working on, including M9140, EBC129, and Pfizer. So we have great ADC platform for troubleshooting existing unmet medical needs, and then we like to aim for the first FDA-approved CEACAM5 ADC. Thank you very much. Thank you for your great presentation. So Q&A session will be conducted at the end of Ms. Chang Soo-kyung's presentation. So we did joint research, secured those data sets. So now we'd like to have a Q&A session. So the reason why we did CEACAM5 high was that all the pharma companies want to deal with as many patients as possible. So I told about 20% response for the high and 5% for moderate expressors. But actually, the efficacy of a drug itself is very important, intrinsic efficacy. But we need to consider various combinations of factors such as patient's compliance. In the clinical settings, the eye infection, ocular toxicity is very serious matter we need to consider. So those horrible adverse events, it would be paper talks. But in terms of hands-on management, that is one of the adverse events that is very hard to deal with from the perspective of doctors. So we need to consider high as well as moderate expresses in order to expand indicators. So not just payload differentiation, but also antibody differentiation would also be important. Thank you very much for the answer. Let us move on to the second question. Regarding MMA, that was developed by Singapore's A-Star. So the low incidence of neuropathy. MMA has about 50% of incidence of neuropathy. We use different ADC platform. It does not mean that we can overcome the payload-related toxicities completely. For example, prior researches. For example, actually, the treatment durability matters. Because as for the neuropathy, when the medications are they administered for a long term, there would be negative impacts. So for patients with shorter treatment durations, there is less incidence of adverse events. That is also related to the MMAE payload-related adverse event. So as long as we can minimize non-specific payload release, then I think that is very manageable when we use ADC platform. I think that is about Merck Sequen-5 ADC. When TOPA is used, then hematology cannot be avoidable. As long as we have that payload, ADC platform can be enhanced with link stability. In this regard, I think that LigaChemBio's the platform is great. Yeah, that is the answer. So we do not have additional questions. So if you have any question from the floor, please raise your hand. Actually, we have another question pre-registered. So I'd like to ask for your patience. We are currently typing the third question. So your questions from the floor are welcome. What is the main mechanism of ocular toxicity that is related to payload, not related to target? That is because DM4 was utilized. Why DM4? Because that is characteristics of that payload. When I consulted with LigaChemBio, so I told that payload selection is very important, and the LCB is doing great about that. Any additional questions from the floor? If you have, please raise your hand. If there is none, we'd like to conclude the session with this. Thank you very much.
Now, would like to invite the Center of Immuno Anti-Cancer Center, Dr. Jang Soo-kyung. She's going to present on when LCB stings cancer. virgin chemistry, linkers, and immunology. She has over 22 years of experience in immunology, and she is in charge of developing next generation drug. We are learning a lot about ADCs. In this session, I'd like to take you to a different landscape. You might be a bit tired and dozy. I'd like you to wake up and join me in this new adventure. Let me talk about cancer immunotherapy. The existing chemotherapy and target therapies are directly attacking cancer cells and killing them, but immunotherapy is activating immunocells, and these immune cells are controlling cancers. As you see, there are many different therapies, not only for cancer but also for other diseases. Merck's Keytruda is a PD-1 antibody-based immunotherapy. It's very well known, and it is recording top sales over the last few years. And also this year is going to see the #1 sales in this drug. And also, for when you see the drug is one of the key metabolic diseases, and it is a driver disease in the pharmaceutical industry, and its prevalence is increasing, and PD1 is very good, but its response rate is like 20 to 30%. So we need a drug that can complement the Keytruda or the Porter drug. So we are trying to develop the complementary drugs. Keytruda was approved in 2013, and it has many side effects, and many research has been done. As a result, at clinics, such side effects has been handled better than before. So the tumor microenvironment, so we have more understanding about it. And given this practical situation, immune checkpoints and many different immunotherapies are being developed. For example, STING and cytokine modulators. And these modulators are being attached and combined with antibodies. And also biospecific antibodies are being developed. So there's a need for breakthrough anti-cancer drugs, and we are actively researching on that. LigaChem, you might be curious about LigaChem developing immunotherapy, why we are doing this, and if we do so, what type of immunotherapy we are developing. You might wonder about this. In a way, LigaChem is developing immunotherapy. It's not a weird thing. Keytruda has been used at clinical settings for decades, and there have been thousands of combination therapy trials. And as a combination therapy, chemo drug is most effective. As you know, ADC is a conjugate attached with chemotherapy. So maybe we can add ADC, then it will be very effective drug. It will create very great synergistic effects. So LigaChem's chemotherapy and chemistry and linker technologies, they can be combined to target IO. So we are developing such a payload like this, and this development strategy is associated with LigaChem's Vision 2030. So in terms of expanding our payloads, so we are developing many different types of payloads. with many different mode of actions. So IO drugs and other MOA payloads and toxin payloads can be conjugated. By doing so, we can create a new modality. So it is also in line with expanding modalities. So ultimately, ADC is very effective, but we want to deliver this great efficacy to as many patients as possible. So we are trying to develop combination therapy with ADC. And also, we want to test and prove the efficacy of combination therapies. So it is also in line with our directions of reducing toxicity. So we focus on STING in applying our technologies. STING is a protein that our cells have. STING is attacking outside attackers like viruses or bacteria, or if our immune or cells are sensing this is something foreign body is not normal, that kind of sensing is happened by the sting. So the sting is actually triggering the immune cells to activate immunity. So in terms of cancer, it might be a little bit complicated, but to give you a nutshell explanation, let's say in a cancer cell, there is a abnormal cell, and that information is communicated to our immune system by STING. And STING also activates the immune system, and it also activates immune cells. So if it combines with the anti-cancer drugs, it can actually maximize its effect, anti-cancer effect. It's called STING agonist. So LigaChem is developing STING agonist. So based on its proprietary chemistry and linker technology, we are trying to develop STING agonist in the form of anti-conjugate or small molecule. That way we are trying to secure our competitiveness. I think I skipped one important part. I think I will discuss it later. LCB39 is a program involving STING agonist small molecule. So we are developing a STING agonist small molecule, and we aim to IND stage next year. STING agonist LCB39 is a systematic administration IV drug, and our highest priority is on safety, and we actually conducted a toxicity test in monkeys, and we secured substance that can actually ensure safety when it's administered. Although LCB39 is a synthetic, but it is a specific characteristic of being accumulated within cancer. So a STING agonist can actually convert that. Let me give you more details about LCB39. LCB39 is a direct form STING agonist. And its LGB's beta glucuronide trigger is attached. It's conjugated like that. So it is increasing the soluble quality, and as it's attached, its penetration into cell has been reduced a lot. So although it's administered systematically, it has very high safety. Although it's safe for the whole system, as I said earlier, LCB39 has a characteristic of being accumulated within the tumor's cells. So it's a very rare characteristics, but LCB39 has this property. Additional property is that the enzymes expressed in tumor and so it actually activates LCB39, and also it increases active the metabolite. So with this, we can actually catch two birds with one stone. This is the data. Let me briefly explain to you as easy as possible. This is the in vivo efficacy result. In many mouse models, we conducted in vivo efficacy. So it's a dose-dependent, dosage-dependent cancer control effect has been very excellent. And we see the cancer cell doesn't grow with this. So basically, immune memory has been created. This is another important point. This graph might look so complicated, but don't be overwhelmed. Let me easily explain it to you. This model is called ART, lung cancer model. So in the lung, we planted tumor, and then we administered the LCB39, and we see this great result. So we actually extracted a cancer cell, and we actually proved whether LCB39 has the anti-cancer control. So when you look at the red bar, it shows really big difference. So the T cells are much activated, and T cells can be activated, and many T cells can be created by the relevant cells, and the number of those cells has increased a lot. And the cancer cell eating cells have been amplified as well. So we see the result. The LCB39 has increased the environment from cold to hot. This is the data that proves that. This is another differentiating point of LCB39. We really want to highlight this. So we administered LCB39 to the mouse with tumor at 3 different doses, and then we extracted blood and a normal organ cell, and then we wanted to test whether there is the active metabolites or LCD39 are found. So we actually observed that the metabolites and LCD39 were present in those cells. So we conducted further research. So the blue line is the amount of a drug that can actually activate the metabolite. So as I said earlier, metabolites were observed, and the dosage that can actually activate metabolites is the red line. So 1 milligram kg. So we see a lot more amount here, and even at a less dose, we were able to detect it. So the amount of LCD39 is doing its job. So according to this data, we were able to verify that. This is a combination potential. This is a very important part that is specific to our company. We are conducting many different combination trials, particularly ADC and PD-1 combination trial. This is the Dato DHC and Trux ADC 29. So you see the LCB, when LCB 39 is administered in combination, as you see in the red line, the result is very homogeneous and the performance is really excellent. As you see in this data, LCB 39 can be utilized in combination with the immune checkpoint or the ADC drug, and it can actually deliver more benefits to patients and also offer treatment opportunities to patients. And this kind of potential market is really possible. So you might ask this question. So pharmaceutical companies have been developing drugs using STING agonist, but there's no success case in clinical settings. There exists such a challenge. So in my company, we look at this in two perspectives. First, compound itself. As I said earlier, LCB 39 has very differentiating characteristics. Although it's a synthetic compound, it can be selectively accumulate within tumors, and the normal cell toxicity has improved a lot. So such characteristics are being shown by our drug, and it offers really great potential. Another thing is that clinical translation. Existing pharmaceutical companies do not have a special clinical strategies or differentiating strategies. Although it's a great drug and great target, but you need to also have a great clinical strategies. So when you conduct clinical trials, you need to have really great strategies. And what we are also focusing on biomarkers. What kind of patients are really responding to our drug? So we will focus on biomarker studies. Currently, we are actively pursuing biomarker studies. So the clinical designs and with this drug, and we are doing our best to become the best in class and first in class. Now, let me focus more on ADCs. By utilizing STING agonist, let me talk about our anti-conjugate. So you learned a lot about LigaChem's link technologies and the ADCs. You see the immune modulator here. You can actually apply it to AIC. So the site-specific characteristic can actually precisely target, and this technology applies to immune modulator as well. And also, I mentioned that we are expanding modalities. So ADIC is one of them. So ADIC has this form of drug. How many toxins does it have? How many immune modulators does it have? So we can actually accurately create these based on our LigaChem core technologies. One example is that STING agonist DAR4 target A drug. This is one example of AIC with those characteristics. So this is the translation model. See, you can see the dose. It's a dose dependent, and it shows really great efficacy. Effective molecules for STING agonist are well expressed, and you can see that in molecule STING agonist AIC. This is target B ADIC with DAR2. So target B is less expressed in this model. The red colors have two different concentrations, and their efficacy is limited. But although with the same DAR, but compared to the same dosage of competitor, it has a better efficacy. And if you have a higher dosage, we can see the complete remission. So when you look at the STING agonist used in ARC and AIDC, they have a different structure compared to LCB39. So we chose the most optimal payloads, and this is the most optimal STING agonist. The AIC and AIDC, as I mentioned in those areas, so it has the optimal pair and optimal combination, especially ADIC. Within one drug, there is DUI present at the same time, so it creates a synergistic effect. So there's a certain MOA, and when you have two drugs with different MOAs, its potency increases. So in this combination, because they have two different MOAs, so memo function and also the immunity activation, so it has very long-term efficacy. This is the combination of ADC and IO. At LigaChem, not only STING agonist, but also IO and direct T-cell activating payloads are being developed. So there are areas where there's some needs that couldn't be addressed by the existing ADC, so we are developing next generation ADCs. So hopefully our this development strategy will bring about the next generation ADC, which is the best in class. Regarding the presentation by Dr. Jung, Dato ADC and PD1. So we actually did a Dato ADC because we wanted to show the better efficacy compared to the existing drugs in market. So we are going to open a Q&A session. Dr. Jung and Mr. Jung, would you please come up to the stage? I think it would be better for me to stand up so that I can have a better view of the questions on the screen. So would you please load the first question? DAR 1 and low DAR was mentioned in this question. We increased the therapeutic index. All ADC's therapeutic index is affected by the linker and payload as well as targets. So as we pursue low DAR, we can actually increase number of targets where PBD can apply. So we can actually expand our market in the new area. That's why we are pursuing low DAR. Currently, our drug is safe, but by lowering the DAR level, we can actually increase the safety even further. By doing so, we can apply our drug to the indications where the existing drugs cannot target. Second question. Earlier, I briefly mentioned about this. There's no complete clinical design developed yet, but we are focusing on biomarker, and we are trying to identify the patient groups who show the drug, prove our drug activity. So we are trying to develop relevant biomarkers regarding that. So we are going to do the clinical design on top of that, but we are trying to identify the STING agonist efficacy and toxicity. That way, we are trying to design our clinical trial. Thank you very much. Third question. We have quite many questions. You are comparing the monoclonal antibody and the biospecific antibody. Although you are targeting the same thing, depending on the epitope or affinity, it could actually develop different toxicity. So although it's a biospecific antibody, what kind of target combination, that matters, and how you can adjust the affinity for each target, and what kind of biospecific antibody platforms you're going to use. Depending on that, its efficacy and toxicity might differ. So we don't just simply developing biospecific antibody. So we need to modulate and adjust the affinity and the number of payloads and what kind of platforms we are going to use. We are looking to many different aspects. So although biospecific antibody has general benefits, we are not just pursuing them, simply looking at those benefits. CMCs might have challenges, so there's more steps involved in that. So it actually increases the production cost. In that respect, the existing biospecific antibody platforms can be utilized. Based on my experience, this is my insight. Next question. This question is quite long. Regarding this, we often receive a question like this. This is about a STING target. STING is activated chronically. It can be actually pro-tumor. So this kind of signal might actually go towards pro-tumor direction. So the question is about that. In a way, even in the academia, there's no clear definition about this. However, when you look into the literature, you can find out the following. Chronic pathways and signalings are activated. Inflammation can happen. So it can serve the role of the stage where the cancer is being created. But most patients we see are suffering from already advanced and aggressive cancer. So they are not at the stage of developing cancer. They are not on the initial stage. STING activation versus drugs. So when you compare the two, they are different. When you look at the non-clinical testing, small amount STING agonist versus a high dose STING agonist, when you look at the cancer reaction, it's all dose dependent. So it is different from the cancer initiation pathway. So the STING agonist therapy at the stage of treating advanced cancer is different. The cytokine release can be strengthened. Although it's the same STING agonist, the amount that releases a cytokine or the amount that does the opposite, they're kind of close, but we found the STING agonists, well, of which window is quite wide, and also dosing schedule period. So we don't do it frequently, just once every week we administer it. So we administer it and then give time for the immunity to be recovered. So I believe we can actually adjust it by spacing and scheduling it properly. So we need to change the way we think. IL-6. I think that the person who raised this question knows a lot about STING. So when STING is activated, the inflammation pathway can be turned on, or there's another pathway, but our drug, we don't know why, but our drug is cytokine release syndrome. So we don't see that CRS pathway, and we see the pinal dominant interferon pathway. We see type interferon pathway more with our drug. Another question? Another question about STING? This is a great question. In the biomarker studies, this area is included in our biomarker study. Do I need to explain more? The existing pharmaceutical companies who developed STING agonist didn't consider this, but beta glucuronidase, that is our specific one, and they didn't consider this part. Biomarker and study designs will incorporate this, which will be differentiated from the existing clinical trials by other companies. So we are not in an opportune time to answer that question. Regarding this is what we are actually doing right now. We conjugate IO payload to antibody. That way we can have more activation in tumor. So we are trying to add more drugs to fight cancer. So you talked about differentiation. I don't get it clearly. So what cells the antibodies are targeting? So that could be a differentiating point. The LCB39 is a chemistry drug, but it can be injected into, it can be delivered to a tumor a lot, and it also has a properties of the antibody conjugate. So our STING agonist could be different in terms of what type of cells it is targeting, and also it's different from the general STING agonist because it is targeting cancer, and it brings more STING agonist to cancers, and it is exposed when it is exposed to the entire body, the systematic toxicity can be reduced. Next question. Mr. Jung can answer this question. Target affinity. I think you're talking about target affinity. With a higher target affinity, that doesn't mean that internalization is being done well. So neutral pH affinity and acidic affinity, depending on that, toxicity and efficacy might vary. And in terms of pharmacokinetics, on rate, off rate might vary depending on whether it is the liquid cancer or solid cancer. So for solid cancer, off rate is more important. And the within tumor cell infiltration rate matters, but the movement into the tumor is also important. So proper balance is important. So cellular infiltration and the tissue infiltration, you need to modulate those rates for optimization. Next question. The existing targets, for example, HER2 and TROP, and they couldn't find the accurate biomarkers, HER2 positive or these things are based on Herceptin and also IHC. So you need to have a different you can actually need to have a different standards. So for novel target, selection of biomarkers is very critical. So when we started developing, we already started selecting biomarkers, but when we select targets, we already start a research on the selection of biomarkers. So if it is a novel target, biomarker is more important. The last question, and then we are going to take a break. Last question. This method you mentioned in your question is really troublesome for us. You need to actually do it, and you need to pursue both depending on indication, although it is the same target. Sometimes ADIC might be more appropriate. For example, ADIC might be more effective for breast cancer. For other disease, ADC might be more effective, and after that, immunomodulation could be given. So you need to take a personalized approach. You don't just choose one. You can actually develop both to a certain degree. Basically, internally, in the past, best in one class ADC for one target, but now we are trying to form best package for the relevant target. So we are trying to form the package drugs for the same disease. So we can actually cover all relevant diseases with the package. Because we are trying to develop such package, it takes a lot of time, and also there's more research projects being underway. Let me give you more explanations with a focus on ADIC. ADIC, you might think ADIC might not be different much. The two good drugs can be used in combination, but those two drugs can be loaded onto one modality as one drug, and its advantages are being really clear. So compared to the combination therapy of those two, if you add them onto one modality, it shows some differentiating effect in terms of toxicity and efficacy. So BAC and the STING agonist can be loaded onto one modality, and it creates a really great synergistic effect, which is very attractive drug. So in the second part, we talked about new payloads, strategies, and ADC pipelines, and also STING-related discussions. So this concludes the second part, and at 3:10 we are going to start part 3. Thank you very much.